is very high, and okay.
Sorry, that was me. I forgot to record, and I have a lot of people asking for the recordings, Francois.
Okay,
My bad. You don't have to
okay
restart. I'll just apologize to all the people listening. We're a couple minutes into the intro.
Sorry, you want me to restart?
No, no need.
Okay. Okay, so I was just mentioning the blood-retinal barrier that prevents many things from going into the retina or out of it unless there is an active transport. The photoreceptors have a very high energy consumption, probably the largest or highest energy consumption of any cell type in the human body, and that may be due in particular to the dark current that photoreceptors generate, and perhaps also to the continuous membrane synthesis that they need to renew their outer segments. And then, of course, one of the questions that I'm particularly interested in is we have a selective vulnerability of photoreceptors in many retinal diseases, and could this be related to energy metabolism? So another little recap, the main textbook biochemical pathways that most of you, I'm sure, have heard about, the glycolysis and the Krebs cycle. So in glycolysis, we have glucose going in. It's a 6-carbon atom molecule, and it's split into 2 pyruvate molecules, which each have 3 carbon atoms. And that liberates 2 ATP molecules and 2 NADH molecules per glucose. So this pyruvate, textbook knowledge has it that it goes into the Krebs cycle and then oxidative phosphorylation. And if this runs on completely, then the pyruvate will be completely oxidized to carbon dioxide and water, and this will liberate 4 NADH, or will generate 4 NADH molecules, 1 FADH and 1 GTP molecule. And each NADH and FADH molecule can be converted into roughly 3 ATP molecules, so that you get roughly per pyruvate about 16 ATP equivalents out of it. So this is what every textbook has, I believe. What the textbooks are sometimes a little bit not so clear about is that glycolysis is, well, it's inefficient, that's obvious, in terms of ATP output, but it's also really fast. And the Krebs cycle, on the other hand, is efficient in terms of ATP output, but it's very slow. It's about 100 times slower than glycolysis. And this is a problem because these 2 pathways are coupled by pyruvate. So this coupling requires basically that the 2 pathways run at the same speed. And that is not the case. So how does this work? You could, of course, conceive that you can have, for each glycolytic unit, you can have like 100 Krebs cycle units, and then maybe this could work. But if you have a situation like in a photoreceptor or in any other neuron, where you have a rapidly changing energy demand, then you run into a problem as soon as you switch from very high consumption to low and back to high consumption. And so this is a problem for the coupling. And then the other thing that, of course, is important to consider is that glycolysis happens in the cytoplasm and the Krebs cycle in the mitochondria. So there's also a transfer of metabolites required. So the idea that I want to put forward today is that this coupling, pyruvate coupling, does not really happen. And I know it has, every textbook has it, and I would say it's not really happening, at least not in neuronal cell types, and probably not in photoreceptors either. And why do I think like that? I think to understand this, we have to go back in the evolution, and very far back, actually. So this diagram here goes from present time, 0 to 4.5 billion years ago, when our planet Earth formed. And the interesting thing is perhaps that the last universal common ancestor, LUCA abbreviated, this critter lived already on this planet about 4.2 billion years ago. So already 300 million years after the formation of the planet, we had the first forms of life, our last universal common ancestor. And the atmospheric conditions at that time on Earth were very different to what it is today. And notably, the atmosphere was rich in hydrogen, or in other words, it was a reducing atmosphere. And this, of course, influenced metabolism that could evolve under these conditions. And this is exactly when glycolysis evolved, which is perhaps the first metabolic pathway to ever evolve under reducing conditions under high hydrogen atmosphere. So about 3.5 billion years ago, some bacteria evolved, cyanobacteria, or related to today's cyanobacteria, that evolved oxygenic photosynthesis. So they released oxygen. However, in the Earth's crust, there were a lot of sinks for oxygen, so that the oxygen levels in the atmosphere, they remained basically zero for a long period of time, until we reach about 2.4 billion years ago, perhaps, a thing that is called the Great Oxygenation Event. And this is the first time that oxygen rises in the atmosphere. So the atmosphere turns from a reducing atmosphere into an oxidizing atmosphere. And this dramatically changes how life can perform metabolism. And it is here at around 2.2 billion years ago that oxidative phosphorylation evolves. Oxidative phosphorylation is basically photosynthesis turned the other way around, except that, that wasn't possible as long as there was no oxygen in the atmosphere. Only when there was enough oxygen in the atmosphere could you do this. And then later on, we have another important event. We have this endosymbiosis, where some of these bacteria that could perform oxidative phosphorylation became incorporated into bacteria that were predominantly glycolytic. And this is the first time that these two pathways came together, glycolysis and oxidative phosphorylation, and this gave rise to eukaryotic organisms. Eventually, when the oxygen levels in the atmosphere rose higher, above 10% approximately, we saw the rise of multicellular organisms and, well, eventually even us. So the point that I would like to make is that oxidative phosphorylation and glycolysis for a very long time have been separated, and they didn't evolve in the same organism. They evolved in separate organisms, and they came together in a rather artificial way. And this created artifacts that we can still see in metabolism today. And I would like to bring to your attention a few of those. The first one was discovered around about 1857 by Louis Pasteur. Louis Pasteur, famous French scientist. He was contracted by the French winemaking industry, who wanted to make alcohol production more efficient. And he studied this, and he actually came up with a very satisfying solution for them. And he found out that oxygen actually drives energy metabolism towards the Krebs cycle and oxidative phosphorylation. And vice versa, if you reduce oxygen levels, then you can drive metabolism towards glycolysis. So if we apply this to the retina, highly perfused tissue, highly oxygenated, so you would expect the retina to use oxidative phosphorylation, according to Pasteur at least. Now, about 100 years ago, Otto Warburg came along, and he studied cancer metabolism, and he found out that metabolism normally, this is early days, glycolysis hadn't been elucidated fully, but he found out that glycolysis happens in most organisms. The glucose would be split into pyruvate, which would enter oxidative phosphorylation. So he was after cancer pathogenesis, and he studied many different tumors, and he found that in tumors, the pyruvate that is resulting from glycolysis is converted into lactate and does not undergo oxidative phosphorylation, even though there is oxygen present. And he called this later on aerobic glycolysis. Now, in his studies of different tumors, he also found that the retina was the only healthy tissue that behaved very similar to a tumor, that was also releasing lactate, producing a lot of lactate, even in the presence of oxygen. So this is kind of an enigma that may be puzzling us to this day. Why does the retina behave like a tumor when really from its energy demand and from the availability of oxygen and perhaps other considerations, you would think it shouldn't do that? So the key points that Otto Warburg found out here is that the retina uses the rather inefficient glycolysis, even though it has oxygen available. He calls this aerobic glycolysis. And this, of course, is a clear contradiction to what Pasteur found. Shortly after Otto Warburg, another gentleman came along, Herbert Grace Crabtree, and he picked up Otto Warburg's studies, but took it a little bit further and combined it with Pasteur, kind of. But he, instead of varying oxygen, he varied glucose levels. And what Crabtree found was that high glucose levels can cause a shutdown of the Krebs cycle and oxidative phosphorylation, so that high glucose levels drive glycolysis. So this is kind of a complement to the Pasteur effect. So what does that have to do with the retina? So I have to talk a little bit about another, well, problem. I mentioned that this glycolysis and oxidative phosphorylation, they're kind of kinetically incompatible. They're thought to be coupled by via pyruvate, but kinetically this doesn't really work. And I'm not the first one to find this out. Actually, it was, I think, for the first time shown in the 1960s. And to solve this dilemma, a couple of gentlemen from Switzerland, Luc Pellerin and Pierre Magistretti, in the early 1990s came up with an idea that would split glycolysis and oxidative phosphorylation across two different cell types, across astrocytes and neurons. And that way solving the kinetic incompatibility. So if we look at what would this mean for the retina, this is called the astrocyte neuron lactate shuttle hypothesis. And for the retina, this would mean, you know, astrocytes, there are no astrocytes in the outer retina, but there's Müller glial cells. So the Müller glial cells would somehow have to take up glucose that comes from the bloodstream. They would have to convert it into lactate and then feed that to the photoreceptors, and then the photoreceptors would use the lactate to do oxidative phosphorylation, if that hypothesis was transferable to the retina. Now, more recently, Jim Hurley in his lab came up with an alternative idea, where they found in this paper in eLife in 2017 that the RPE cells would pass glucose on. Indeed, glucose would go through the RPE cells and it would go into the photoreceptors, and the photoreceptors would perform glycolysis, not oxidative phosphorylation, glycolysis, and then the lactate released would fuel oxidative phosphorylation in the RPE cells, in a nutshell. So these are two, you know, alternative views that one could have about this. And we tried to study this. And one thing that I particularly found puzzling is that the inner segments of photoreceptors, they harbor a lot of mitochondria, which are, you know, supposed to do oxidative phosphorylation. So if this hypothesis was correct, then why would the photoreceptors still have mitochondria? If they were only performing glycolysis, they wouldn't really need the mitochondria. So the first thing that we looked at was at the expression of glucose transporters. And what you see here to the left are cross-section through a retina, where glucose transporter 1 is labeled in magenta, and it's strongly expressed in the RPE cells. Green is a co-labeling with rhodopsin. And yeah, we find it very strongly expressed in the RPE cells. Another glucose transporter that we looked at is glucose transporter 3, which we find to be strongly expressed in the inner segments, actually at the border between inner segments and outer segments of the photoreceptors. And here is a rhodopsin co-labeling to illustrate where the GLUT3 expression is localized. So this setup is very interesting, where we would have GLUT1 in the RPE cells and GLUT3 in the photoreceptor cell, photoreceptor inner segment even. This matches the setup that we have in the brain, where we have GLUT1 sitting on the astrocytes, and then GLUT3 sitting on the neurons behind the astrocytes. And what is interesting about is that both GLUT1 and GLUT3 are high capacity transporters, but the affinity to glucose is lower for GLUT1 and higher for GLUT3, so that you have a decreasing gradient of glucose from the blood vessels towards the neuron, matched by an increasing affinity of the glucose transporters. And that allows for an efficient transport of glucose through the tissue. So this would be supportive of strong glucose consumption and high glucose transport and into the photoreceptors. So what we then did, we looked at different markers for different retinal cell types. For instance, we stained RPE65 as a marker for RPE cells in the retina. Glucose transporter 1, also labeling RPE cells predominantly. Then here we have pyruvate kinase M2, that catalyzes the final step of glycolysis. We have cytochrome oxidase, a key enzyme of oxidative phosphorylation, and we see both of them expressed very strongly in the photoreceptor inner segments. We then used retinal explant cultures, where we take a mouse retina and put it into a culture dish on a transwell insert. And we can do this in two different ways. We can either culture the retina with the retinal pigment epithelium attached to it, or without the retinal pigment epithelium, so that we have different experimental conditions with RPE or without RPE. We can block glucose transporter 1 with a compound that is rather selective, 1.9-D-deoxyphosphorylation. We can block PKM2 with another selective inhibitor called chiconin. And we can uncouple oxidative phosphorylation using this drug called FCCP. So we can manipulate these targets that are expressed in specific cell types of the retina. And what we get, or the first thing that we analyzed, was how does this affect cell survival? How does this affect cell death? So we performed the famous Tunnel assay for dying cells, shown here in light blue. To the left is a control retinal explant culture that was not exposed to anything. Then a culture without an RPE. We have a slight elevation of cell death because the RPE is missing. When we block glucose transport with 1,9-D-deoxyphosphorylation, we get more cell death. When we block PKM2 with chiconin, more cell death. And when we block oxidative phosphorylation, even more cell death. And we can quantify this as the percentage of tunnel-positive cells in the outer nuclear layer. And you see that there is indeed, each of these drugs has each of these treatments has a significant effect. And FCCP really is not good for retinal health.
Francois?
Okay, so now, of course, here in the outer nuclear layer of mouse retina, we know that about 97% of the cells of the photoreceptors here are rod photoreceptors. So how about the cones? To assess that, we did a cone staining, cone meristin staining in the control retina. And then in the no RPE retina, we get a decrease in the cone numbers. With the 1,9-D-deoxyphosphorylation, the glucose transport inhibitor, we get basically no more cones left. With chiconin, the cone number decreases. With FCCP, it doesn't seem to be affected. We can quantify this, and this is indeed what we see. So if we compare the control as the baseline to the GLUT1, glucose transporter 1 inhibitor, this is really deadly for cones. Whereas FCCP, the drug that abolishes oxidative phosphorylation, or that abolishes ATP production from oxidative phosphorylation, this doesn't affect cones really at all. But with FCCP, we have a very high percentage of cell death. So those cells that are dying here, they're not cones, so they must be rods. And over here, we have some cells dying, and all of the cones are gone. So this is probably mostly cones. And the conclusion that we can draw from this is that rods appear to be reliant on oxidative phosphorylation, whereas cones really seem to need glycolysis. At least they really seem to need glucose transporter 1 on the RPE to be functioning. So afterwards, we took the retina from these retinal explant cultures and performed a metabolomic analysis for these different treatments. And this is now put sideways. And we looked at a number of different metabolites and color-coded the relative concentration of these metabolites in the different experimental conditions. And then there was a Ward's algorithm used to cluster these metabolites into this colorful map. We see these nice patterns, and then we try to interpret this, which unfortunately turned out to be far more difficult than I had originally anticipated. Well, another thing that we did is we performed a principal component analysis, looked at whether principal component 1 and 2, how they behave, plotted them against each other, and found that the 5 experimental conditions, control being one of them, are really, distinct to each other. So the treatments, the different treatments really change the metabolomic patterns that we observe. Now, to try and analyze this in more detail, we use different algorithms and artificial intelligence-based algorithms and quite a few different bioinformatic tools, but unfortunately, none of them revealed anything meaningful. So we ended up going through this by hand for each metabolite and each experimental condition. And I want to show you just a few examples in the interest of time. So glycolytic activity, and how does this relate to the Krebs cycle? So of course, glucose is split into pyruvate, and this can be converted into lactate in aerobic glycolysis, or it can enter the Krebs cycle. Now, the Krebs cycle is one of the key metabolites that comes out of it is GTP, which really can only be produced in significant amounts by either the Krebs cycle and a specific enzyme, succinate ligase, or it can be generated from excess ATP by an enzyme called NDK. But that requires that you have an excess of ATP. So what we can now do is we can plot the ratio of GTP over lactate in the different conditions, and then look at what that shows us. So in the control, we have in the control condition, we have a certain ratio, GTP over lactate, which we can take as normal. If we culture the retina without RPE, this ratio goes down. So there's less GTP, more lactate, indicating that more glycolysis was performed. With the 1,9-D-deoxyphosphoglycan, so the glucose transporter inhibitor, it goes in the other direction. So it goes into the direction of more oxidative phosphorylation, more Krebs cycle activity. With chikunine, it's not really changed. But with FCCP, it really goes down, also in the direction of more glycolysis, which is kind of logical because under FCCP, the retina cannot use oxidative phosphorylation at all. The only pathway that remains to generate ATP is then glycolysis. Okay, so this is one way of looking at it, but it's perhaps not the best way of analyzing this rather large data set. So another thing that we looked at is the glucose consumption in the medium in which the retina was sitting. So we have these different conditions. Unfortunately, we didn't have a very good standard for measuring glucose in the medium. So this is arbitrary units. So the control has a certain level of glucose in the medium. No RPE has less, indicating more consumption. With the glucose transporter inhibitor, we have more glucose in the medium, so less glucose consumption. Chikunine, perhaps not really changed. FCCP, it looks like there's more glucose consumption.
What was the starting
more
amount
glucose
of glucose
versus the control. We can also look at lactate, and this is perhaps more interesting. Also here, normalized concentrations. In the control, there is a little bit of lactate release, indicating some aerobic glycolysis being performed by the retina. In the no RPE condition, the lactate release increases. With the glucose transporter inhibitor, it decreases. With the PKM2 inhibitor, it decreases. With FCCP, lactate concentration rises, so indicating more glycolysis. Okay, so this is in the medium. So now we can still we can look at the metabolites in the retina, in the tissue. Oh, okay, sorry. Now, this thing here is a little bit odd. When you look at how much lactate is liberated by the retina that doesn't have an RPE, and one idea could be that this is caused by the Crabtree effect, by an excess of glucose. And I will come to that in a couple of more slides. So now we can look at different conditions, different experimental conditions individually and compare them. For instance, we compare the control condition versus the no RPE condition and look then at different metabolites, each of them individually. So for instance, we can look at citrate. And citrate is high in the control and low in the no RPE situation. And the enzyme that produces citrate synthase, is strongly expressed in the inner segments of the photoreceptors. There's also some expression in the inner retina, but predominantly in the photoreceptors. Another metabolite that we could look at is lactate, obviously. And the enzyme that is producing lactate is LDHA, and it's strongly expressed in the photoreceptors. So compatible with photoreceptors performing glycolysis and producing lactate. And another interesting metabolite is alanine. And the enzyme that can produce it is alanine transaminase, which we find to be strongly expressed in cone inner segments, as shown here by a co-labeling with conerestin. It's also expressed in the inner retina in bipolar cells, but in the outer retina, it's only expressed in cone inner segments. And what is interesting is that alanine and lactate, they behave exactly reciprocal to each other. In the control, we have a lot of alanine and no lactate. And in the no RPE retina, we have no alanine and a lot of lactate. So what pathway could be behind this? So another group of metabolites that we look at is this here, leucine, isoleucine, valine, branched chain amino acids, which are also interesting for energy metabolism because they can be used as fuels in oxidative phosphorylation. So what I would like to suggest, a little bit provocative perhaps, is that this lactate, this strong rise in lactate, it only appears in the retina that doesn't have an RPE. And this is also the situation that Otto Warburg used and that many others after him have used to assess lactate production in the retina. So could this be an artifact of the fact that the retina doesn't have an RPE? I think that's at least something that one can discuss. So let me remind you about two important pathways, the Corey cycle and the Cahill cycle. So the Corey cycle was discovered in the 1930s by Getty and Fred Corey. And this Corey cycle has it that glucose from the bloodstream is taken up in muscle. It was originally discovered for muscle. And in the muscle, the glucose is split into pyruvate that generates NADH and ATP. The NADH is then used to reduce the pyruvate to lactate. The lactate is released to the bloodstream and then enters the liver, where the lactate is again used for glucose to create pyruvate, and then this pyruvate goes into gluconeogenesis, and the glucose is re-released into the bloodstream, closing the loop, closing the cycle. Now, this was thought to be very important for energy production in the muscle, but actually in the 1960s, Mr. Cahill came along, and he found that actually more important is another cycle, which was later on called Cahill cycle. And here, glucose is also split into pyruvate, but then instead of using the NADH to reduce the pyruvate to lactate, instead glutamate is used to transaminate pyruvate to yield alanine and alpha-ketoglutarate. The alanine then goes back to the bloodstream, and then the alanine is used in the liver to regenerate pyruvate, and this is used for gluconeogenesis, the glucose can then go back to the bloodstream. So the key metabolites of these two pathways are lactate or alanine. And if you see one or the other, you can say which pathway was being used for energy production. So we found a lot of alanine. Now, another thing that is interesting about Corey and Cahill cycle is that they're independent of mitochondrial respiration. For the Corey cycle, this is pretty obvious. glucose converted to pyruvate, split into pyruvate converted to lactate, and you don't need the Krebs cycle and oxidative phosphorylation at all for this. Now, in the Cahill cycle, it is not like that. So you have glutamate entering the cell, then pyruvate together with glutamate is transaminated to give alanine. Pyruvate becomes alanine, glutamate becomes alpha-ketoglutarate, and the alpha-ketoglutarate can then enter the Krebs cycle at this level, this lower half, so to say, of the cycle. This can then be processed to oxaloacetate, and the oxaloacetate can be converted back into pyruvate using pyruvate carboxykinase. It actually goes via phosphoryl pyruvate to pyruvate. So what is interesting here is, in this case, you don't need glycolysis. You can run this independently of glycolysis. And you also don't need the upper half of this Krebs cycle. And this is important because this upper half, the first 3 steps of the Krebs cycle, are the very slow rate-limiting steps of the Krebs cycle, which really, slow it down. If you run only the lower half, the whole thing, the remainder of the Krebs cycle gets to be significantly faster, about 5 to 7 times faster than the full Krebs cycle. And that is important for cells that need energy fast. So, for this to work, you need pyruvate carboxykinase, because otherwise you would get stuck at this level with all the oxaloacetate. So you need pyruvate carboxykinase. And where's the enzyme that does this? So pyruvate carboxykinase 1, the cytoplasmic form, is strongly expressed in raw photoreceptors in the outer retina. And pyruvate carboxykinase 2 is exclusively expressed in cone inner segments. So in cone inner segments, in the mitochondria, this pathway, this Cahill pathway, could be run pretty efficiently and rapidly. So if we now look again at this metabolite map and look at all these different metabolites, what else is interesting? So another metabolite is, of course, aspartate, and perhaps its counterpart is glutamate. And again, they behave exactly reciprocal. And this may bring to mind the glutamate aspartate shuttle, perhaps. But I think what is behind this pattern is a different thing. So first of all, where's the enzyme expressed? Or is the enzyme that would be able to use glutamate and aspartate, where is it expressed? And is it expressed in the retina? And this enzyme is called aspartate aminotransferase. Some people call it also, or abbreviated, we abbreviate it here AAT. Other people abbreviate it AST or ASAT, also sometimes GOT. So that unfortunately doesn't make it easier. At any rate, AAT1 and AAT2 are both expressed in the retina, and they are both expressed also in photoreceptor inner segments. So they could be used to convert glutamate to aspartate. And then we have another interesting metabolite here, which is N-acetyl aspartate, which is very interesting, and I will come to that on the next slide. Because of this. So we're proposing that the retina is not really using the full Krebs cycle, but that it may be using an abbreviated mini-Krebs cycle that has been described for the first time in the early 1990s. So in this mini-Krebs cycle, glutamate would enter the mitochondria. AAT, aspartate aminotransferase, would take the amino group from glutamate, put it onto oxaloacetate, converting it into aspartate and generating alpha-ketoglutarate. The alpha-ketoglutarate can then go through this lower half of the Krebs cycle to generate oxaloacetate. Now, if you run this on continuous glutamate input, you will be facing the problem that all this aspartate will be piling up, and this will clog up the works. So you need to get rid of the aspartate. But unfortunately, that's not membrane permeable. So how do you do this? Well, you can acetylate it, and the acetylation will make it membrane permeable. And then you get N-acetyl aspartate. This is a metabolite that we see very prominently in the retina. So if you do this, you can also enter not only glutamate, you can also enter branch chain amino acids, either at the level of acetyl-CoA or at the level of succinyl-CoA. And this will generate additional reducing equivalents or GTP. And if at some point you would ever run out of the acetyl-CoA, then you can use pyruvate carboxykinase to regenerate pyruvate and generate acetyl-CoA from this. So you have kind of a double loop that is intertwined that allows you to generate, to run this lower half of the Krebs cycle on glutamate and branch chain amino acid input, with the output being N-acetyl aspartate. And yeah, and again, this upper half of the Krebs cycle with the rate limiting steps, you wouldn't need those. Okay, so, and this again can be done independently of glycolysis. So if we look at this in well, as a summary, kind of a summary together, we might have in the photoreceptor, in either the rod or the cone, or separated in across these cells, 2 or 4 different, up to 4 different pathways. The first one would be the classical, where you have glucose entering glycolysis, converted to pyruvate, and then the pyruvate goes through the full Krebs cycle. The classical textbook thing. The alternative would be the Gaur cycle, where the pyruvate is converted into lactate, and this does not use oxidative phosphorylation. The second alternative would be the Cahill cycle, where you would generate alanine and could run oxidative phosphorylation in addition. And the fourth would be the mini-Krebs cycle that would generate N-acetyl aspartate and run on input of glutamate and branch chain amino acids. And the interesting aspect of this is that that except for the first pathway, the classical biochemistry textbook thing, all other things, you can run them in parallel and independently of each other. And now, how would this work out in terms of energy yield for these different pathways? How much ATP could you generate from this? So in the classical setup, you would have glycolysis running in the cytoplasm. Glycolysis would generate 1 NADH and 1 ATP per pyruvate molecule. Then the pyruvate could enter the mitochondria. And in the full Krebs cycle, you would generate up to 4 NADH, 1 FADH, and 1 GTP. If you run the Gaur cycle, so the aerobic glycolysis exclusively, what you get out of it is only 1 ATP per pyruvate molecule. So this is really not very good in terms of ATP output per carbon input. The Cahill cycle, this would be relatively interesting because the NADH that is liberated is not used. And also in the mitochondrial part, you can still use this to generate 2 NADH, 1 FADH, and 1 GTP molecule. So it's quite a bit more efficient, especially when you convert the NADH and the FADH into 3 ATP approximately each. And then the mini-Krebs cycle, which is a bit complicated because it runs on glutamate and branch chain amino acid input. It could generate up to 4 NADH, 2 FADH, and 2 GTP. So it might actually be, depending on how the stoichiometry works out, it might be more efficient in terms of ATP output per carbon input than the classical biochemical setup. Okay, so this is how much ATP you can get per carbon input. But another important question is how much ATP can you get per time? And I mentioned that in the beginning that for a neuron and a photoreceptor, the timing might be more important than the actual yield. So what is plotted here is ATP output per time, arbitrary units. And if we take the Gaur cycle, so the conversion of glucose to lactate as an example, we get 1 ATP out of it. And we just say arbitrarily, this is 1 unit of time, whatever timing that is. And how would that behave towards the full Krebs cycle? where pyruvate is converted to carbon dioxide, or glucose is split into pyruvate and then converted to carbon dioxide. Well, this would be about 100 times slower. It would yield more ATP, but if you calculate the rate, ATP per time, then the full Krebs cycle is really not very attractive. So if you're a neuron and you've just fired an action potential and you need to repolarize your membrane, like now in the next couple of milliseconds, then this is really not very good. As efficient as it may be in terms of ATP output per time, it's not good. The Cahill cycle, which is much faster because it doesn't have the rate limiting steps of the full cycle, of the full Krebs cycle, is faster and generates more ATP per time. And the mini-Krebs cycle running on glutamate and branched chain amino acids may produce even more ATP per time, even more than the Cori cycle. So that's it for the timing of energy output. And this ordinate here shows, like, you know, what would deliver the same amount of ATP per time as glucose, and what would be better and what would be worse. And with that, I'm actually getting to my summary. So I would like to say that photoreceptors, they are very likely to use both glutamate, branched chain amino acids, and glucose as their fuels. Our data indicates that rods need primarily oxidative phosphorylation. Cones are more likely to be dependent on glycolysis. Rods may be using the mini-Krebs cycle. There's some evidence for it that I unfortunately didn't have time to go into, but including the expression of certain enzymes that you need for it. Cones are more likely to use the Cahill cycle, and perhaps in addition to it, in parallel to it, the Cori cycle. What is interesting is that lactate, alanine, and N-acetyl aspartates, they are kind of signature metabolites. When you see them, you can deduce what kind of pathway was running to produce energy. And this is a drawing that a PhD student, a former PhD student of mine, Yi Chen, has produced, illustrating or summarizing this, where we think that the rods may be using the mini-Krebs cycle, liberating N-acetyl aspartate, notably, whereas the cones are more likely to use the Cori cycle, liberating lactate, or the Cahill cycle, liberating alanine. Now, of course, this is only the beginning of, I think, what will be many studies into energy metabolism. And we think this is this versatility of different mechanisms, different pathways, may allow the cell, the photoreceptor cell, to match the yield and the rate optimally to the momentaneous metabolic needs, which are dramatically changing on a millisecond timescale. And when it comes to open questions, there are so many. So for instance, the kinetics and the stoichiometry of the different pathways is still a little bit sketchy. Modeling of these molecular pathways and the kinetics of it would be very interesting to do, to get perhaps additional insights. Then eventually, would these different pathways, would they allow us to explain the different vulnerability of rods and cones and different diseases? So I'm working often predominantly on retinitis pigmentosa, where we have rod genetic defects, and then there's a secondary cone degeneration that nobody really understands why it happens. Does it perhaps have to do with the energy metabolism and some sort of metabolic support from rods? Then the molecular basis for the Crabtree effect. The Crabtree effect is the shutdown of oxidative phosphorylation when there's too much glucose. Nobody really understands how high glucose levels shut down oxidative phosphorylation. Still open. And of course, eventually, if once we understand these things sufficiently well, will we be able to use it to rationally design novel therapeutic interventions that target energy metabolism, that increase energy production in the cells that need it, or perhaps reduce it when they don't really need it? So this was my summary, and I would like to close now by acknowledging the people that were involved in the study, also cooperation partners and the funding, and there's some recent publications and preprints that maybe you want to have a look at. And other than that, I'm very open for questions.
Can you hear me now? Hello?
Can you hear us, Francois?
Yeah, we can hear you.
No,
Yeah.
I cannot hear him. Sorry. Sorry, my speakers were off.
Yeah, we were trying to talk during your talk. We're trying to
yeah,
ask
I was
questions
wondering,
in
there
the
was
middle.
no, and it's noise canceling, so I really didn't hear anything.
Yeah, no worries. So I think we have a lot of questions in the chat, but perhaps we'll give people who have something on their mind a chance to answer first. And I think the first question that I have is just kind of a fundamental, do we really know for sure that the retina does want to optimize energy production? Or is that just a sort of a guess based on the fact that there are a couple experiments showing that it can sort of use energy rapidly? Or maybe, you know, just aerobic glycolysis and lactate production is good enough.
I'm not sure I understood your question. So you were asking whether the... Yep.
How do we know that the retina needs to use pathways that are more optimized to generate ATP than sort of a conventional textbook biochemistry pathway? I'm not saying you're wrong. I'm just saying, you know, is there any evidence to believe that the retina wants to optimize speed or ATP production?
Just to be clear, I'm not saying that, you know, this is what I just exposed here is the full truth. You know, it could very well be wrong. But I think the data that we have matches this interpretation so far. So, but anyway, so why do we think, or why do I think that the retina needs to adapt its energy production? So first of all, we know that the photoreceptors, they have an extraordinarily high energy consumption. This is shown independently by other people before. And we also know that cones have an even higher energy consumption than rods, so approximately 2 times more ATP consumption per time than rods on an average basis. And also we know that the energy consumption or the energy demand changes dramatically from light to dark. So in a matter of milliseconds, the energy consumption goes down when you go into the light, and then it goes up when you go into darkness again. So that must be handled somehow. And how exactly is it handled? I think this is what I'm trying to get at here.
Okay, anybody have any questions they want to ask? Maybe if you unshare, I can sort of see people.
Okay, I can, yeah, sorry, I can do that. Oops. Yeah, I thought Jason Miller was moving his lips, but I couldn't hear him.
We can't hear you, Jason. I don't know if you're talking.
Still can't hear you, Jason.
But you
have
have
at home, you can come to my office, Jason.
we have questions in the chat also while people are sort of thinking, oh, Jason typed something out maybe. Okay, yeah. Well, why don't you, did you have access to the chat, Francois?
Yeah, I just need to.
You can probably ignore like the first 3 boxes.
Yeah, just a second. I need to shuffle around some windows on my screen. Yeah, now I see them. Yeah, so first one is, I would guess the high level of perfusion available photoreceptors are somewhat countered by the rather large diffusion distance across outer segments. Yep, that's possible, but I cannot really comment on it, I'm afraid. I haven't
Yeah, that
measured
was
this
just when
over.
you're talking about, the sort of very high supply of oxygen. I agree, the choriocapillaris is, extremely fast flow. Of course, there's going to be a large oxygen supply, but whether or not that is sort of as accessible as it would be if the cells that consume the oxygen were right next to that supply, that's, I guess, the part that I was saying was less clear to me.
And then EW, Eric, I guess, what is the diffusion limit in distance for oxygen? Not sure I can answer that either. Probably not. But what I know is that oxygen is extremely fast in diffusion. So I used to work in stroke research, and I used to try and get cell cultures to be non-oxygenated. And as soon as you would, open the lid for like a split second, the liquid solution, water-based solution would be fully oxygenated. So it's really very difficult to get something to be not oxygenated. But in neuronal tissue, I think the key thing is the consumption. If you have a very high consumption, then of course you can get a tissue anoxic, like for instance in the brain after a stroke, and that can happen very, very quickly. But if you didn't have the consumption, then the diffusion is not a limit. Okay. Actually, now I see that Daniel, you actually answered that. Sorry. Nancy, was your glucose transporter 3 antibody characterized? We performed Western blot, and the band size matched what our expected molecular weight was. But that was the characterization of it, to be honest. For the GLUT1, we did the same thing. We actually also went a little bit further. We tried like, I think, 6 or 7 different GLUT1 antibodies, because the question was a little bit, perhaps a bit more controversial. So we tested a number of different antibodies, some of which gave very, very different results. And the one that we finally went with, we subjected that to an IP pull down and analyzed that by mass spectrometry. And we found this to indeed pull down glucose transporter 1. So we think that the one where I showed the images of is really specific to glucose transporter 1. We didn't do that kind of characterization for glucose transporter 3. Not yet, at least. Mohita, Gaur, what is the age of mice for GLUT1 and PKM2 immunohistochemistry? That was postnatal day 15 in the image that I showed. And that was the same for the retinal explant cultures. So we, I probably forgot to mention that, we explanted the retina at postnatal day 9, then kept it in culture for 2 days without any treatment to adapt to culture conditions, and then treated it for 4 days with the different drugs. Or in the case of no RPE, it was 6 days without an RPE. Okay, then Daniel, I think, does the presence of RPE block O2 transit to the tissue? I would expect not, but I think the consumption would be a problem. So if there was a major consumption of oxygen, then that would limit, of course, the amount of oxygen that could reach the retina.
Well, yeah, I mean, theoretically, the retina is consuming oxygen the entire time, and you're adding on a tissue, so that kind of blocks to a greater extent the ability of oxygen to get there. But it also blocks other things. I mean, I think the problem, one issue that I would love if people solved for ex vivo cultures is we don't have a way to perfuse the choriocapillaris. If we could, that would be amazing.
Yeah, I think people have done that in the 1970s, the 80s, and perhaps as late as the 1990s. I think there's some experiments. I think it was a gentleman called Niemeyer, who took a live cat, took out the eye, anesthetized cat, took out the eye, and then hooked onto the eye, outside the animal, and a perfusion, and then measured a variety of interesting things. But unfortunately, I don't think he did so much metabolic studies. It was more into functional analysis of the retina.
Mhm.
So studies like that nowadays are very difficult to get approved.
Yeah, well, one thing I did want to note while I have the mic is that Doug Dean has done these studies in anesthetized pigs, living anesthetized pigs, and he has kind of done these sort of arteriovenous metabolite measurements for a few different things. So, he shows, for example, that there's a time of day effect that affects glucose consumption and also lactate production. And that means there's more venous lactate coming out of outer retina circulation than coming in. So I think, there's definitely some argument to be made for there's at least some rate of in vivo lactate production that eventually escapes the circulation.
Yeah, even in our explant cultures, we do see, so the medium that we supply the retina with, which is a medium free of antibiotics, free of serum, is entirely defined, and there's no lactate in it. So when we take it after 2 days of cultivation of the retina, even in the control condition, we do find lactate in the medium, and so it has to come from the retina. But it's just not as much as we get, not as much lactate as that we get from a no RPE retina, for instance. So then, were the inhibitor treatments titrated to ensure maximum effectiveness? Yes, for chiconin and for the 1,9-D-deoxyphoscolin. For FCCP, we didn't do it because there was so much previous literature on it. So here you ask, some of these inhibitors have off-target effects. FCCP can depolarize the plasma membrane. Yes, I'm aware of that, but at the concentrations that we use, this shouldn't be an effect that should be detectable. And if it did that, if FCCP did in fact depolarize the plasma membrane, then it would cause, cell death rather rapidly, definitely over the 4-day culture time frame that we have here. And we haven't seen cell death with FCCP treatment in any other cell than rod photoreceptors. So if FCCP had a general plasma membrane depolarization effect, then all the cells in the retina should be dead all over. And that was not observed. So this, I think, indicates that the concentration that we used was low enough to not cause such an effect. In the glucose assay, what was the starting concentration? Yeah, this is a little bit of a tricky question. As I briefly mentioned, we don't have an absolute quantification of this because the way we did the metabolomic measurement, we couldn't quantify it absolutely. So it's a relative quantification. We are now working on this and are trying to get to an absolute concentration, but it's not. We should probably do a new experiment with a new set of data to be really sure about this. The starting concentration in the medium, this is maybe interesting, is 19.1 millimolar. So that's a relatively high concentration. When you would compare that to diabetes, for instance, where you would think in the bloodstream, concentration above 7 millimolar would probably be pre-diabetic, above 10, certainly diabetic. Here we use 19, but in culture, where we don't have a blood flow, we routinely need to increase glucose concentrations in order to allow the cells to get enough glucose. Another consideration may be the consumption of glucose, because our culture, that we change the medium every 2 days. So there needs to be enough glucose to last for 2 days. So at any rate, the starting concentration was 19 millimolar. Then there is another question, John G. Is it possible that the high lactate from no RPE group results from no RPE consumption of lactate? I think we cannot exclude that. I don't think so, but can I really say that with any certainty? Probably not. I would think that the RPE doesn't consume so much lactate, but I may be totally wrong here. No, so this is a possibility. However, what I can say, if you vaguely remember this graph where we had these 5 different conditions: no control, not consuming, not producing so much lactate, no RPE producing a lot of lactate, and the FCCP, so the blocker of oxidative phosphorylation, produced almost the same amount of lactate, indicating that there is a change in the metabolism, similar to a loss of oxidative phosphorylation in the no RPE retina.
Well, the funny thing is, FCCP should accelerate, for the most part, the electron transport chain activity, and perhaps thus the TCA cycle. So you might not expect I mean, you could expect that the cells respond by inhibiting carbon entry into their mitochondria because they realize that their mitochondria aren't producing ATP, but it's also quite possible that the mitochondria are very active.
Yeah, but at any rate, the cell cannot produce any ATP from when it's exposed to FCCP. So the only pathway that remains is glycolysis. And then how do you get rid of all the pyruvate? There's basically no other way than to convert it to lactate. Even if you have oxygen. So then the next question is: malic enzyme Me2 could be an alternate to PEPCK, to pyruvate carboxykinase. Yes, although I'm not 100% sure how this would work, but I have seen this, and I always wanted to look into this possibility, but didn't get around doing it yet. That's no question. Christian Grimm, is GLUT3 expression detected in rods in single-cell RNA sequencing studies? I'm not aware of such studies where they show GLUT3 expression. I don't want to be too blunt, but I'm not a big fan of single-cell RNA sequencing studies. Especially not for low abundance transcripts, where there is a propensity to detect either too high or too low mRNA. Another problem with single-cell RNA sequencing is that when you dissociate the cells, which is what you do here, you get a very rapid change in the transcriptomic pattern. And there's a recent paper by Raju Rajala in, I think, October last year, where he used a different methodology to freeze the tissue, the retinal tissue, before the dissociation, and then dissociate only the frozen cells so that the RNA profile wouldn't change. And he found significant changes induced by, even a very short dissociation and even a very short period where the cells remain as single cells. Another point is, of course, that mRNA, and I cannot repeat this often enough, does not equate protein expression. Many people especially in the genetic field, they tend to think that if you look at mRNA, that's enough. And no, it's not enough. You need to look at the protein to be able to say anything with certainty about what's happening, and then perhaps whether that protein is active or not.
Can I comment on this a little bit? So one thing,
sure.
It's you're completely right. This is Dorota. You're completely right about this assumption in omics field. However, now a lot of people are working on single cell proteomics. So fortunately, we will have some snapshot on that. I just mentioned in the chat that I suggest single nuclear RNA-seq from frozen tissue. So you freeze it, snap freeze, and then you don't dissociate anymore. You just isolate nuclei. And not only you get only the snapshot of current transcription. So there's no accumulation in the cytoplasm, but also you have no more transcription ongoing because everything happens on ice. It's fantastic. I strongly recommend for people that want to look at snapshots of transcription information, transcriptomic information. Not just how the transcriptional program is in the cell, you know what I mean? And with all the accumulated mRNA here and there, rather just to look what cell is doing right now. And I can assure you, we can easily dissociate the cell types with this method as well.
Okay, that's very interesting, and yeah, I think it goes well with this study from Dr. Rajala from earlier last
absolutely.
year. Yeah. So then, okay, this is a comment on the GLUT3. Christian saying that GLUT3 protein enrots only in very special situations. Okay. Then Alison, why do you think that photoreceptors die without RPE in vivo, but that wasn't the case in your ex vivo experiment? Yeah, that's a good point. So they, in the in vitro culture situation without RPE, the viability of photoreceptors is reduced. And actually, this was also shown here. You saw that the rate of cell death in the no RPE retina was increased compared to the control retina with an RPE. So the retina with RPE, we can routinely culture for 4 weeks or up to 6 weeks, actually, without a major loss of any of the neuronal cell types. If we culture the same retina without an RPE, the viability is strongly limited to maybe 12 to 14 days. But you really see a loss of photoreceptors as you go along. It's like almost like an inherited retinal degeneration when you have no RPE. So it does conform to the in vivo situation where you get photoreceptor loss without RPE. Okay, and then the final comment from Gaur, I guess. Okay, so then I think this is the questions from the chat, if I'm not wrong.
Yeah, you got them. Nice job. Nancy has a question.
Thank you for all the questions.
For the chat. They're not done. Nancy has a question.
Yes.
Oh, this is sort of just a comment. Really nice talk. But we've done RNA scope with looking at SLC 2A1 and 2A3 for GLUT1 and GLUT3. And clearly we show that GLUT1 is in the outer retina, including the RPE and Mueller cells, and GLUT3 is limited to the inner retina. And we use the GLUT1 antibody we've validated in knocking out GLUT1 from RPE, rod photoreceptor cells, Mueller cells, and actually lens. And the labeling matches the knockout. So I think it really clearly shows where GLUT1 is expressed in the retina. And so I think there are specific antibodies and right, the recent paper from Christian Grimm's lab showed that GLUT1 under certain circumstances, GLUT3 was increased in photoreceptor cells. And so we didn't see that when we knocked GLUT1 out of the retina with the CRX-CRE, but we do, it seems that there may be an increase in GLUT3 in the rod delta when we knock GLUT1 out of rods with an inducible CRE from Stephen Zhang.
Yeah. So I've seen your work, obviously, and I find it very elegant. For the RNA scope, it's the same question that we had. Does mRNA indicate protein? Only to some extent. What I can say about the antibody, of course, each antibody has its own character, and sometimes you find things with one antibody that you don't find with the other. So with the methods that we use, we find a very strong expression of GLUT1 in the RPE, and that conforms to your study, I think, rather well. When you look at the images that you can obtain with this GLUT1 antibody at very high gamma levels, then you actually see a staining that is reminiscent of Müller cells. So we didn't do a co-labeling, but it's, you know, it's the only cell type that spans the entire retina. And it does seem to have a little bit of GLUT1 immunoreactivity, much, much less than what we see in the RPE, but I wouldn't exclude that possibility. So yeah, as far as photoreceptors go, you know, I cannot comment further. It's I don't have a better answer than anybody else, perhaps.
I think Allison, you have a question?
Yeah, so we have we've mentioned multiple times how the metabolism is different based on the time of the day and the light conditions. When people are doing these experiments with RPE and retina in culture for long periods of time, like weeks, is there a light component that's added? Has anyone tried that? And what, if they have, what are the results of that?
Yeah, this is a tricky question. So we keep our retina, you know, our explant cultures in an incubator, and there is no light in the incubator, so they're constantly in the dark, except for the, you know, couple of minutes that they are taken out every 2 days to change the medium. How does that influence the thing? We haven't really looked into it, but you're absolutely right. I would expect this to have an influence on the metabolism. I mean, obviously in the light, you would expect there to be a lower consumption, generally speaking, of photoreceptors, because they wouldn't need the dark current. So they wouldn't need to run the dark current. But then on the other hand, there may be, you know, if you did this properly, you might have a diurnal rhythm, and you might have outer segment membrane synthesis, which lipid production, which does consume a lot of energy. So there are two factors in here that could be that are not so easy to disentangle, and we haven't done it yet, and I don't know whether anybody else has.
All right, great question.
Dan, this is Jason. I have one. I have one two clarifying questions. François, the transwell setup is such that everything is laying on top of the transwell, and you have the same media on the basolateral and apical side. Is that correct?
Yeah, in principle. So the retina sits on a membrane, a polycarbonate membrane, and the membrane is positioned on top of the medium, such that at the lower side you have the medium, and on top of the retina you have only a very thin film of liquid that is created by the surface tension of water. So this allows the retina to be very nicely oxygenated. And if you put the retina, if you plunge the whole thing into the liquid, then you actually, I call it drowning. The retina actually dies very soon when you do that. As I said, we can keep it we can keep it viable and functional for weeks at a time if we do it like that, with the retina at the interface between liquid and air. If we put it in the liquid, it lasts for a couple of days at max.
And so all the media you're collecting then is from the basal lateral chamber.
Yes.
Right. And
how do you, there's no other chamber.
Right. Yeah. And how do you keep the explant sort of wet enough that it doesn't dry out on the apical side? Is just capillary action enough, or do you have to literally every day drop a little bit on the capillary actions enough to keep it? Okay.
Yeah. That's fine. You just have to pay attention that the level of the liquid doesn't drop below the membrane. Then the thing dies very quickly.
Oh, I see. So you're controlling the amount by putting how much is in the basal, so that it's just the amount in the basal is in equilibrium just above. Got it. Okay.
Yeah.
And the other question is on your control versus no RPE metabolite heat map. Remind me, red is that there's more metabolites present, correct?
Yes.
So
Red is more, blue is less. Sorry, I should have mentioned that.
That's okay. But so, for example, when you're postulating the BCAA contribution to the mini-Krebs cycle, for example, I'm trying to understand because the BCAAs are higher in the no RPE condition.
Yeah.
So wouldn't that suggest that it's the RPE that's sort of using up the BCAAs and not making them available for the, I mean, the mini-Krebs cycle may be all true, but like there's multiple options to feed into the mini-Krebs cycle, like you said.
Yeah.
How do you interpret that data that I just said?
No, you're right. This is a possibility, and in fact, I think there are several publications that raised or that indicated that the RPE can feed on branched chain amino acids. So this is similar to the question before. You know, how do we know that the lactate is not, the superfluous, the excess lactate in the medium? How do we know that is there because it's not consumed by the RPE? Couldn't that be the same for the branched chain amino acids? Yes. However, when you compare with the other, we have 3 more experimental conditions. The indications are all that the branched chain amino acids. So for instance, in the FCCP condition, where we have the RPE, but no oxidative phosphorylation, the branched chain amino acids are also down, which indicates that they're consumed somehow, perhaps not for oxidative phosphorylation. So to be honest, I don't have a full answer to your question.
No. Thank you.
I think Nancy had another question, if I see correctly.
Oh, your hand was still raised. Anyway, I think unless anybody has any other questions they want to ask, I think we're good.
That was awesome. I mean, like, amazing introduction to.
Right. Thank you so much. Yeah, for the talk.
I would like to thank everybody for asking these questions because, well, we're in a process of trying to get this published, and all these questions, as nasty as they may sometimes be, I like them because they may help us moving forward. And also, I would like to say that, this is one interpretation of the data that we have. There's for sure other interpretations. And I hope that this will stimulate more research into it and that we will get the answers to the questions that I couldn't answer now and on all the other questions that weren't even asked in the future. Thank you.
All right. Thank you. I'll see you next week, everyone.